Hello students, and welcome to the lecture on Deviation from the Basic Pedigree Patterns of Inheritance. The main objectives of today's lecture are to give a general idea of Mendelian pedigree pattern, to explain various patterns of deviations from pedigree, discuss non-penetrance, variable expressivity, pleiotropy, late onset anticipation, genetic heterogeneity, uniparental disomy, to explain spontaneous mutation, to discuss X inactivation and dosage compensation. To begin with, let me first of all give a brief introduction about pedigree. Pedigree is a chart showing a record of inheritance of certain traits for two or more ancestral generation of human beings or domesticated animals in the form of a diagram or family tree. In pedigree, symbols represent people and lines represent genetic relationships. However, in real-life situations, various patterns of inheritance deviate from the basic Mendelian patterns, and we will discuss few of them here in today's lecture. Let's begin with the first part of topic, that is the non-penetrance. Non-penetrance by definition is the failure of a dominant condition to manifest itself. By definition, a dominant character is manifest in a heterozygous person and so should show 100% penetrance. However, many characters fail to express themselves in a generation which would otherwise be expressed as a dominant character. Very often, the presence or absence of a character depends on normal circumstances on the genotype at one locus, but an unusual genetic background, particular lifestyle, or maybe just chance that the occasional person may fail to manifest the character. Now, we will discuss another deviation from pedigree, that is variable expressivity. Expressivity quantifies variation in a non-binary phenotype across individuals carrying a particular genotype. Expressivity therefore characterizes non-binary qualitatively or quantitatively the extent of phenotypic variation within a particular genotype. Expressivity is measured only when there is 100% penetrance. Variable expressivity occurs when a phenotype is expressed to a different degree among individuals with the same genotype. For example, individuals with the same allele for a gene involved in a quantitative trait like body height might have large variance, some are taller than others, making prediction of the phenotype from a particular genotype alone difficult. Further, the expression of a phenotype may be modified by the effects of aging, other genetic loci, or environmental factors. Next is the pleiotropy. The term pleiotropy comes from the Greek pleion, meaning more, and the tropos, meaning way. Thus, a pleiotropy means more change. A pleiotropic gene causes more than one phenotype to change. Pleiotropy occurs when one gene influences two or more seemingly unrelated phenotypic traits. Even though a structural gene may have many end effects, it usually has only one primary function, that of producing one polypeptide. This polypeptide may give rise to different expressions at the phenotypic level. Thus, pleiotropy describes the genetic effects of a single gene on multiple phenotypic traits. It's not essential that all the traits are equally influenced. Sometimes, the effect of a pleiotropic gene is more evident in case of one trait, known as a major effect, and less evident in case of others, known as the secondary effect. Here, I would like to give certain examples of pleiotropy. Number one is the phenylketonuria. A classical example of pleiotropy is the human disease phenylketonuria. This disease can cause mental retardation and reduced hair and skin pigmentation and can be caused by mutation in a single gene that codes for the enzyme phenylalanine hydroxylase, which converts the amino acid phenylalanine to tyrosine. Depending on the mutation involved, conversion of phenylalanine to tyrosine is reduced or ceased entirely. Unconverted phenylalanine concentrates in the bloodstream and can rise to levels that are toxic to the developing nervous system of newborn and can cause effects such as mental retardation and abnormal giant posture. Because tyrosine is used by the body to make melanin, an important component of the pigment found in hair and skin, the failure to convert normal levels of phenylalanine to tyrosine results in less pigmentation being produced, causing the fair hair and skin typically associated with phenylketonuria. The second example is sickle cell anemia. Sickling of erythrocytes occurs due to mutation in the beta chain of hemoglobin. It not only causes hemolytic anemia but also has the following phenotypic effects. Number 1, the skilled RBCs are destroyed by the liver, causing hemolytic anemia. The phenotypic effects of this anemia include physical weakness, below average development, and hypertrophy of bone marrow. The second one is the skilled cells cause the interference of capillary blood flow by clumping of the odd-shaped cells, resulting in damage to every major organ. The individual can suffer pain, heart failure, rheumatism, and other effects. Another case of deviation from basic pedigree is the late onset. Genetic conditions are not necessarily congenital. The genotype is fixed at conception, but the phenotype may not persist until adult life. In such cases, the penetrance is age-related and referred to as late onset. Huntington's disease is a well-known example. Delayed onset might be caused by slow accumulation of a noxious substance, by slow tissue death, or by inability to repair some form of environmental damage. Hereditary cancers are caused by a second mutation affecting a cell of a person who already carries one mutation in a tumor suppressor gene. Depending on the disease, the penetrance may become 100% if the person lives long enough, or there may be people who carry the gene but who will never develop symptoms no matter how long they live. Now, we shall discuss some dominance problems. According to Mendel's law of dominance, when a pair of alleles or allelomorphs are brought together in F1 hybrid, then only one of them expresses itself, masking the expression of other completely. Here, under dominance problems, we are supposed to discuss the exceptions to the principle of dominance. The first one is incomplete dominance or partial dominance or mosaic dominance. When a dominant allele does not mask completely the phenotypic expression of the recessive allele in a heterozygote, then blending of both dominant and recessive traits takes place in F1 and F2 heterozygotes. This phenomenon is known as incomplete dominance. In such cases, the blending occurs only in the phenotype of the F1 heterozygotes, and the alleles maintain their individual identities and segregate from each other during gametogenesis. The F1 gametes produce F2 progeny having the phenotypic and genotypic ratios of 1:2:1. For example, in Mirabilis jalapa, there are two types of flower color in pure state, that is red and white. When a homozygous red flowered plant is crossed with a homozygous white flowered plant, the F1 heterozygotes are found to have pink flowers. The second one is the codominance. It's the phenomenon of two alleles lacking dominant recessive relationship and both express themselves in the organism. Example, the alleles governing the Mn blood group system in humans are codominants. Here, three blood groups are possible, M, N, and Mn, and these are determined by the genotypes MM, NN, Mn respectively. Blood groups actually represent the presence of an immunological antigen on the surface of RBCs. People of Mn genotype have both antigens. The third one is the multiple alleles. They are multiple forms of a Mendelian factor or genes which occur on the same gene locus, distributed in different organisms in the gene pool, with an organisms carrying only two alleles and gametes only one allele. For example, ABO blood group system in human beings showing multiple alleles. Human beings have 6 genotypes and 4 blood groups or blood group phenotypes A, B, AB, and O. The last one is the heterodominance. When the heterozygotes have a more extreme phenotype than either of the corresponding homozygotes, that is its homozygous parents, then it is usually referred to as overdominance, superdominance, or heterodominance. For example, there is a heterodominance when the heterozygote between a pair of factors which control size is bigger than any of the homozygote, recessive or dominant. This type of allelic relationship, which implies interaction between the alleles or of these with other factors of genotype, may be found in qualitative characters and especially those such as size, production, vigor, etc., which are of importance in the breeding of animals and plants. Yet another deviation from basic pedigree pattern is anticipation. The signs and symptoms of some genetic conditions tend to become more severe and appear at an early age as the disorder is passed from one generation to the next. This phenomenon is called anticipation. Anticipation describes the tendency of some variable dominant conditions to become more severe in successive generations. Anticipation typically occurs with disorders that are caused by an unusual type of mutation called a trinucleotide repeat expansion. A trinucleotide repeat is a sequence of 3 DNA building blocks, nucleotides, that is repeated a number of times in a row. In some cases, the trinucleotide repeat may expand until the gene stops functioning normally. This expansion causes the features of some disorders to become more severe with each successive generation. Anticipation is common in trinucleotide repeat disorders such as Huntington's disease and myotonic dystrophy, where a dynamic mutation in DNA occurs. Here, it's crucial to mention what trinucleotide repeats are. Trinucleotide repeats are apparent in a number of loci in the human genome. They have been found in introns, exons, and 5' or 3' UTRs. Increased number of repeats influences the overall shape of the DNA, which can have an effect on its interaction with DNA polymerase and thus the expression of the gene. When the trinucleotide repeat is present within the protein coding region, the repeat expansion leads to production of a mutant protein with gain of function. This is the case for Huntington's disease, where the trinucleotide repeat encodes a long stretch of glutamine residues. When the repeat is present in an untranslated region, it could affect the expression of the gene in which the repeat is found, example Fragile X, or many genes through a dominant negative effect, example myotonic dystrophy. Dear students, let me give you some examples of disease showing anticipation. Huntington's disease with CAG repeats, myotonic dystrophy with CTG repeats, dikeratosis congenita with TTAGG, Friedreich ataxia with GAA repeats, X-linked Fragile X syndrome with CGG repeats. Dear students, the next heading is the genetic heterogeneity. Most Mendelian diseases were originally classified from their phenotype, in other words, their clinical manifestations. As molecular causes have been identified for these diseases, it has turned out that in many cases the same or a similar phenotype can result from a different underlying genotype. This phenomena is known as genetic heterogeneity. Thus, genetic heterogeneity is a phenomena in which a single phenotype or genetic disorder may be caused by any one of a multiple number of alleles or non-allele mutations. This is in contrast to pleiotropy, where a single gene may cause multiple phenotypic expressions or disorders. Genetic heterogeneity describes genetic variation from the normal population. Clinically, genetic heterogeneity refers to the disease that results from multiple gene abnormalities. An example is autosomal dominant polycystic kidney disease. Multiple gene abnormalities are seen in disorders such as autism, cystic fibrosis, and retinitis pigmentosa. The next part of deviation from basic pedigree is the uniparental disomy. Uniparental disomy occurs when a person receives 2 copies of a chromosome or part of a chromosome from one parent and no copies from the other parent. Uniparental disomy can occur as a random event during the formation of egg or sperm cells or may happen at early fetal development. Uniparental disomy can be the result of heterodisomy, in which a pair of non-identical chromosomes are inherited from one parent, an early stage meiosis 1 error, or isodisomy, in which a single chromosome from one parent is duplicated, a later stage meiosis 2nd error. In many cases, uniparental disomy likely has no effect on health or development. Because most genes are not imprinted, it doesn't matter if a person inherits both copies from one parent instead of one copy from each parent. In some cases, However, it does make a difference whether a gene is inherited from a person's mother or father. A person with uniparental disomy may lack any active copies of essential genes that undergo genomic imprinting. This loss of gene function can lead to delayed development, intellectual disability, or other health problems. Several genetic disorders can result from uniparental disomy or a disruption of normal genomic imprinting. The most well-known conditions include Prader-Willi syndrome, which is characterized by uncontrolled eating and obesity, and Angelman syndrome, which causes intellectual disability and impaired speech. Dear students, next and very significant condition of deviation from basic pedigree is spontaneous mutation. Here, we will briefly discuss the different types of mutations. As we all know, mutation is sudden but heritable change in the sequence of an organism's genome that gives rise to alternate forms of any gene. It can simply be put as an abrupt change in the genotype of an organism that is not the result of recombination. Spontaneous mutations are those mutations that occur without known cause. They may be truly spontaneous, resulting from an inherent low level of metabolic errors, that is, mistakes during replication, or they may actually be caused by mutagenic agents present in the environment. Spontaneous mutations arise from a variety of sources, including errors in DNA replication, spontaneous lesions, and transposable genetic elements. Now, let us discuss them one by one. So, first type of spontaneous mutation is errors in DNA replication. The error in DNA replication can result from one of the following three causes. Number one, the tautomeric shift. Each of the bases in DNA can appear in one of the several forms called tautomers, which are isomers that differ in the positions of their atoms and in the bonds between the atoms. The two forms are in equilibrium. Formation of tautomer of any base alters its base pairing properties. Example, the more stable keto forms of thymine and guanine and amino forms of adenine and cytosine may infrequently undergo tautomeric shifts to less stable enol and imino forms respectively. Normally, the bases would be expected to exist in their less stable tautomeric form for only a very short period of time. However, if a base exists in the rare tautomeric form at the moment that it was being replicated or being incorporated into a nascent DNA chain, a mutation might result. In tautomeric form, the bases can form adenine-cytosine and guanine-thymine base pairs instead of normal adenine-thymine and guanine-cytosine respectively. The second one is the substitution mutation. Substitution mutation is also known as point mutation, simple mutation, or single site mutation. It's a gene mutation that results from the substitution of one base pair from another is known as substitution mutation. It may be transition, the most common class comprising the substitution of one pyrimidine by the other or one purine by the other, transversion. In transversion, purine is replaced by pyrimidine or vice versa. And the third one is the frame shift mutation. Aberrant replication can also result in small number of extra nucleotides being inserted into the polynucleotide being synthesized, or some nucleotides in the template not being copied. Addition or deletion of base pair that occurs within the protein coding portion of a gene has the effect of shifting the translational reading frame. In majority of the cases, these results in a failure to synthesize a functional protein, thus allowing the mutation to be identified by its phenotypic consequences. Because these mutations cause a shift in the translational reading frame, they are termed frame shift mutations. The second type of spontaneous mutation is the spontaneous lesion. In addition to replication errors, spontaneous lesions, naturally occurring damage to the DNA, can generate mutations. Two of the most frequent spontaneous lesions result from depurination and deamination. Depurination, the more common of the two, consists of the interruption of the glycosidic bond between the base and the deoxyribose and the subsequent loss of a guanine or an adenine residue from the DNA. resulting in significant genetic damage. Because in replication, the resulting epurinic sites cannot specify a base complementary to the original purine. Similarly, depyrimidination is the loss of pyrimidine base. Deamination, it is the removal of an amino group from a molecule. 3 of the 4 nitrogenous bases normally present in DNA, cytosine, adenine and guanine, contain exocyclic amino group. The deamination of cytosine yields uracil. Unrepaired uracil residues will pair with adenine in replication, resulting in the conversion of a GC pair into an AT pair. Deamination of adenine and guanine results in the formation of hypoxanthine and xanthine respectively. Deamination changes the standard base pairing patterns. For example, xanthine pairs with thymine instead of cytosine and hypoxanthine selectively pairs with cytosine instead of thymine. Dear students, the third type of spontaneous mutations are the transposable genetic elements. Some segments in a genome can move quite readily from one place to another. The movement of those elements has effects on the phenotype of the organism, primarily at the transcriptional level. Transposable elements, also called as mobile genes or jumping genes or transposons, are segments of DNA that can move from one position in a genome to another. They were first discovered by Barbara McClinton in 1940s. This DNA segment translocates to other sites in the genome, essentially independent of sequence homology. Insertion repeats into a structural gene can produce a mutant phenotype, often inactivating the gene into which they become inserted. Insertion and accession of transposable elements depends on 2 enzymes, transposase and resolvase. Now, moving to the last part of today's lecture, that is X inactivation and dosage compensation. The X inactivation, also called lionization, is the process by which one of the copies of the X chromosome present in female mammals is inactivated. As nearly all female mammals have 2 X chromosomes, X inactivation prevents them from having twice as many X chromosome gene products as males, who only possess a single copy of the X chromosome. The choice of which X chromosome will be inactivated is random in placental mammals, such as humans. However, unlike the random X inactivation in placental mammals, inactivation in marsupials applies exclusively to the paternally derived X chromosome. It has been demonstrated that in homogametic XX female individuals, one X chromosome gets characteristically condensed and inactivated. Such chromatin material is called facultative heterochromatin, since it becomes inactive in certain part of the life cycle and resumes activity before entering the germline. The phenomenon of inactivation of X chromosome was confirmed by the observation of the Barr body. Dear students, we have discussed X inactivation. Now we will discuss dosage compensation. Dosage compensation is the process by which the expression levels of sex-linked genes are altered in one sex to offset a difference in sex chromosome number between females and males of a heterogametic species. It was first discovered by Muller in Drosophila. Across species, different sexes are often characterized by different types and number of sex chromosomes. For example, XX in human females and XY in human males. In order to account for varying number of sex chromosomes, different organisms have acquired unique methods to equalize gene expression amongst the sexes. Because sex chromosomes contain different number of genes, different species have developed different mechanisms to cope with this inequality. Replicating the actual gene is impossible. Thus, organisms instead equalize the expression from each gene. It's noteworthy to mention that there are 3 main mechanisms of achieving dosage compensation which are common to most species. These include a random inactivation of one female X chromosome as observed in Mus musculus, a 2-fold increase in the transcription of a single male X chromosome as observed in Drosophila melanogaster, and decreased transcription by half in the both of the X chromosomes of a hermaphroditic organism as observed in C. elegans. Now, let us take a couple of examples. In Drosophila melanogaster, X chromosome is not inactivated, but both X chromosomes are transcribed in females. Compensation occurs by making the single X chromosome in male hyperactive. As a result, the level of transcription of the genes carried by the X chromosome is the same in both sexes. It is mediated by the products of male-specific lethal genes. In C. elegans, dosage compensation is achieved by downregulating transcription of genes on both hermaphrodite X chromosomes. Students, that's all about today's program. Hope you have understood and enjoyed. Thank you and goodbye.